This is Jane. She is 6 months old, and we've noticed that she has missed developmental milestones and has lost skills she previously had. She has lost the ability to grasp and touch things. She has difficulty breathing and experiences repetitive hand movements. 12 months later, her development has regressed, or she has lost the ability to speak and walk. She experiences seizures and she suffers from intellectual disability. After consultation with a doctor, she was eventually diagnosed with Rett Syndrome. Rett syndrome is a severe and lifelong neurological disorder that occurs almost exclusively in girls. It occurs in about 1 in 8,500 girls and is typically diagnosed between the ages of 6 and 18 months. Rett syndrome is caused by a loss of function mutation in the X-linked gene MECP2, which is a master regulator of gene expression in the brain. There are over 500 mutations associated with the MECP2 protein that causes Rett syndrome. However, there have been 8 hotspot mutations that constitute more than 60% of cases. As a result, this protein is a transcriptional regulator that can act on a wide range of genes. MECP2 protein is not only expressed at the level of the nervous system, but in all cells of the body. As a result, a mutation in MECP2 has consequences at the level of the metabolism. MECP2 was initially described as a nuclear protein that binds methylated DNA and acts as a transcriptional repressor. Later, it was described as a multifunctional modulator of gene expression and a transcriptional regulator with activating or repressing actions depending on the molecular context. Thus, MECP2 is needed for brain development and acts as one of the many biochemical switches that can either increase gene expression or tell other genes when to turn off and stop producing their own unique proteins. Because the MECP2 gene does not function properly in individuals with Rett syndrome, insufficient amounts or structurally abnormal forms of the protein are produced and can cause other genes to be abnormally expressed as well. Following the identification of MECP2 as the principal genetic factor underlying Rett, enormous effort has been focused on understanding the consequences of brain cells. Within the brain, cellular expression of MECP2 is predominantly detected in neurons, as well as astrocytes and microglia. It is important to note that during neuron differentiation in early life, the MECP2 protein is important in promoting neuron cell differentiation. While astrocytes help to maintain synaptic structure and shape neural circuits in the developing brain, MECP2 expression in Rett represses astrocytic differentiation. As a result, malfunctioning MECP2 disrupts support for neurodendritic maturation, which can help explain delayed mental development in some individuals. While glial cells are normally critical for protecting neurons during development, MECP2 deficient microglia secrete high levels of glutamate, which causes a neurotoxic effect on the postsynaptic excitatory synapse and causes neurons to die. Thus, MECP2 is predominantly involved in the maturation of neuronal function through processes including synaptogenesis, synaptic plasticity, and establishment of functional circuits. As in the case of Jane, Rett syndrome is characterized by typical early growth and development. Clinical symptoms begin to manifest when the patients are between 6 and 18 months old, resulting in cognitive and motor decline. Some of the symptoms are shared with autism spectrum, and therefore the genetic tests help to confirm the diagnosis. Unfortunately, due to the genetic component of the disease, there is currently no treatment for this syndrome. Only drugs in the context of seizures or motor and pulmonary problems can be provided. Due to these complications, life expectancy does not seem to be able to exceed 40 years. There have been a series of recent discoveries within the field of Rett syndrome. At first glance, it might seem that gene therapy could be an ideal treatment. However, this type of treatment brings a great challenge. The expression of MECP2 gene is mosaic type in girls. In other words, there may be more than one cell line with a different genotype from the same zygote. Therefore, some cells in the body may express a normal amount of the gene and not have the mutation. If gene therapy was used, some cells would have an overexpression of the protein, which can cause the duplication of the MECP2. It has been proven that deep brain stimulation improves learning and memory in Rett mice. Thus, in a recent study, authors wanted to focus on a less invasive approach, so they turned their attention to behavioral training. They decided to test whether training can enhance circuit activity and mitigate behavioral deficits in female MECP2 Rett mice. To determine if this treatment is beneficial, they have replicated this type of stimulation in mouse models, which were separated into 4 groups. The first group was wild-type mice, followed by mice models with Rett syndrome without training, followed by mice with Rett model that had early training. In other words, this means they had training before the manifestation of symptoms. And the fourth group is Rett model mice with late training that happens after the onset of symptoms. Classical behavioral tests were used in order to look into their hypothesis. First, to measure the motor activity and coordination of the mice, the rotor-rod test was completed. The Rett model mice who had training for this test before the presence of their symptoms performed better than the other mice in the Rett models. Then cognition was measured with the water maze test. The results had the same tendency as in the coordination test. The mice who had an early training before their symptoms did not demonstrate memory loss until 24 months. This data suggests that early training is beneficial in mice in the Rett model. Then they looked at engagement of task-specific neurons. They discovered that water maze training activates a subset of neurons that encode task-related information. These neurons express genes such as CFOS, which when activated allows synaptic plasticity, which has a huge impact on memory and learning. Because cortical and hippocampal dysfunction underlies the learning and memory abnormalities of Rett mice, they asked whether task-specific neurons in these regions confer the benefits of pre-symptomatic training. They used a CFOS-targeted recombination in active populations, which we'll refer to as FOS trap, to label task-specific neurons activated during water maze training. FOS trap requires 2 transgenes, one that expresses CRE-ER and one that expresses a CFOS with a reporter, in this case TD tomato, in activated neurons. Without tamoxifen, CRE cannot undergo recombination in neuron cells. Administrating 4-hydroxytamoxifen immediately following training ensures that only neurons activated during training are labeled. The results showed that wild-type and Rett mice had an equivalent number of labeled TD tomato positive neurons in the cortex and hippocampus regions, important for spatial memory. In wild-type mice, all TD tomato positive neurons expressed MECP2, but in Rett mice, only half of the TD tomato positive neurons expressed MECP2. This indicates that the loss of MECP2 does not affect the activation of neurons that are task-specific. Further research would be needed to determine what causes the detrimental impact of specific neurons in people with Rett syndrome. To determine if the activity of task-specific neurons are necessary for the beneficial effects of early training, they inhibited their reactivation after training and tested the effect of the spatial memory of mice. They first injected an AAV encoding M-cherry, or the HM4 di-cherry in wild-type mice and Rett model mice. All groups began training at 4 weeks of age and were retested in the water maze in the presence of CNO or control. If task-specific neurons are necessary for the benefits of pre-symptomatic training, preventing their reactivation would impair spatial memory. The results found that neurons expressing M-cherry expressed CFOS, but those expressing HM4 di-M-cherry did not, indicating that CNO or control prevented the reactivation of task-specific neurons. CNO impaired the spatial memory of wild-type and Rett mice expressing HM4 di-M-cherry. Thus, they concluded that the activation of task-specific neurons is therefore necessary for pre-symptomatic training to benefit Rett mice. Their overall conclusion was that early training improves neuronal function. Finally, it is known that when Rett phenotype is reduced, there is improvement in the morphology and electrophysiology of neurons. So they did an experiment to observe in more detail the morphology of specific neurons of CA1 of the hippocampus. The result demonstrates that early training allowed the development of complexity in mouse dendrites that do not express MECP2. In a Rett phenotype, dendrites are usually underdeveloped, and therefore this demonstrates that there is improvement at this level. This type of treatment therefore seems promising. Although this treatment does not eliminate the Rett syndrome, it could improve the quality of life of children with the disease and even delay the symptoms. The fact that early intensive training is safe, non-invasive, and enhances circuit function in multiple behavioral domains makes it an appealing treatment even for infants and young children. Neurogenesis is the process by which new neurons are generated, and as discussed, this process is disrupted in Rett syndrome, which may contribute to the cognitive and behavioral symptoms of the disorder. To better understand the disruption of neurogenesis in Rett syndrome, further experiments need to be performed to develop an understanding of how we can treat this disruption to improve quality of life for patients like Jane. For future experiments, we will be using induced pluripotent stem cells, which are generated by reprogramming somatic cells, such as skin cells, into a pluripotent state, which means they can differentiate into any cell type in the body, including neurons. We will generate the induced pluripotent stem cells from Rett syndrome patients by taking a biopsy of their skin, and differentiate them into neurons in vitro to study the cellular and molecular mechanisms of the disorder. This approach allows us to investigate Rett syndrome in a human cell model, which more closely resembles the human brain than traditional mouse models and allows us to avoid using animals. By using induced pluripotent stem cell-derived neurons, we can study the effects of MECP2 mutations on various aspects of neuronal development and function, such as normal morphology, synaptic activity, and gene expression. Using the induced pluripotent stem cells, we will perform a neurosphere assay. The neurosphere assay involves the culture of neural stem cells in vitro to form neurospheres, which are aggregates of neural progenitor cells that can differentiate into various types of neurons. To study neurogenesis in Rett syndrome, we will culture induced pluripotent-derived neural stem cells to form neurospheres, and then we will compare the number and type of neurospheres generated to those generated from healthy control cells. Differences in the number and size of neurospheres will indicate changes in the rate of cell proliferation and neurogenesis in the patients with Rett syndrome. We can also use the neurosphere assay to test the effects of potential treatments for Rett syndrome by treating induced pluripotent-derived neural stem cells with small molecule drugs or gene therapy vectors. This will allow us to assess the effects on neurosphere formation and differentiation as a result of these treatments. Overall, the neurosphere assay provides a useful tool for studying neurogenesis in Rett syndrome and identifying therapeutic targets for the disorder. By using induced pluripotent-derived neural stem cells, we can investigate the cellular and molecular mechanisms of the disorder in a human cell model, which more closely resembles the human brain than traditional animal models.
Created using Powtoon.